Our group's topic was Ethical Issues of Using Live Animals in Research by Rachel, Jordan, Regina, and Megan. An introduction to our topic of live animal testing begins with the use of animals for research and testing has been around for a very long time, hundreds of years in fact, the majority of the testing being done for human benefit. Most people believe that the use of animals in testing is justified as long as the animal does not suffer unnecessarily. Many medical advances have been made through the use of animal testing. Drug testing is useful in the development of new treatments for infectious and non-infectious diseases. Animals also serve as a tool to understand the effects of medical procedures and surgical experiments. They are used to obtain products like vaccines and antibiotics, which are then used in treatments. Some experiments require certain organs to be tested on and may result in the animal being killed for the researchers to be able to use the organs. Other times, the animal is alive during testing, which calls into question an ethical issue, if the animals have the right against pain and distress. With medical advances, there have been alternative methods created, such as computer methods, which uses computer-generated simulations to predict various possible biological and toxic effects of a chemical or potential drug. Cell and tissue cultures. The cells and tissues from the liver, kidney, brain, and skin are removed from an animal and can be kept outside of the body for a few days to several months or even a few years. Benefits associated with this technique are easy to follow, less time-consuming, and are less expensive. Almost all cosmetics, drugs, and chemicals are tested using these methods, which brings into question, why are animals necessary? Without doing preliminary studies on selected animal species, it would be unethical to test untested chemicals on humans. There are many obstacles to overcome when creating a new drug that only an in vivo testing allows. Doing so in vitro is too challenging. For example, creating an artificial blood-brain barrier or an intestinal barrier is very difficult, and seeing how drugs respond to absorption is difficult to recreate in an in vitro test. To summarize, in vitro drugs are faced with a static system, but in vivo, they are subject to very dramatic conditions. The regulation and history of animal testing. The beginning of animal rights and regulations started in the early and mid-1800s. In 1824, the Royal Society for Prevention of Cruelty to Animals, an animal rights organization, was formed. Later, in 1876, an Act for Prevention of Animal Cruelty was created in the UK. It did not reach the United States until the 1960s. The United States legislature began to look at the issue of animal welfare and animal testing due to a cover page of Life magazine in the 1960s. After much public interest following the cover image of an emancipated dog in a research facility, the Animal Welfare Act, AWA, was created. The AWA covers all species of animals in all types of research, with the exception of birds, mice, rats, and agriculture animals used for production purposes. Many more regulation offices overseeing animal welfare in laboratories, such as Public Health Service. This policy applies to all species and to all research facilities funded by the National Institute of Health. The Office of Laboratory Animal Welfare. This is the agency that oversees regulations. The Guide for the Care and Use of Laboratory Animals, also known as the Guide, was first published in 1963 as a manual for facilities that use animals in research. The latest version of the guide, published in 1996 by the National Research Council, contains standards for the husbandry, housing, veterinary care, personal training, and physical plant specifications for research institutions. In 1885, the AWA was amended to provide for the formation of the Institutional Animal Care and Use Committee. Its purpose is to review research proposals involving animals and to oversee institutional animal care and use programs. The ICOOC must review the institution's animal care and use programs and inspect animal housing and use areas every 6 months. The ICOOC is also charged with reviewing and approving research activities involving animals. Another form of regulation for animal testing was the creation of the 3 R's: reduction, refinement, and replacement. The concept was first discussed in 1957 by Charles Hume and William Russell at the university's Federation for Animal Welfare. This approach motivates the use of minimum number of animals, i.e. reduction, and the total number of animals used in the experiment. The use of animals must be planned and refined carefully in such a way that pain and distress caused during the experiment should be minimized. Furthermore, if possible, animals should be replaced with alternate methods.
So why is the use of live animals for animal research a concern? People often stated that research is only acceptable if pain and distress is minimized. And in a recent web-based survey to explore people's willingness to support the use of mice in chronic versus acute pain research, the majority of the participants oppose the use of mice for either chronic at 68.3% or acute at 63.1%. Secondly, is why animals must be used in the first place. Many studies looked in the use of cellular tissue research to take over the use of animal research. And lastly, concern grows with those of differing backgrounds. So women, those in lesser economic standing, and those with less education are more likely to be concerned with the well-being of animals used in research. Some ethical questions that need to be answered are, do we disregard consent when using animals in research? And how will we know if they are truly in the best living conditions that we can give them? Is this really a necessary practice that enhances the quality of life for the animals as well as the humans? And is the use of some animals more ethical over others, such as mice versus chimps?
So who are the main stakeholders and what are their goals and interests in regards to the ethical issue of using live animals in research? Well, for this question, there are three. First is the scientists. Their interests are to continue their research and to save lives. Their goals are that they want to further their research and make medical breakthroughs. The second main stakeholder is the general public. Their interest is to benefit from cures and treatment that the research will provide them. Also, they would be negatively affected by having untested treatments used on them. Their goals are to want to use the products and treatments that are safe from testing. And the third and final main stakeholder are the animals, generally represented by animal welfareists and activists, and some of the general public. Their interests are that they want the animals to live a natural life free from pain and discomfort. Their goals are that they don't want animals to be used for research. The ethical perspectives are really in two zones. One is the pro-live animal research, which are generally the scientists and the general public. The other ethical perspective is anti-live animal research, represented by animal welfareists and activists, and some of the general public. First, let's look at the ethical perspectives of the researchers and general public, who argue for pro-live animal research. First, they like to claim that animal testing has contributed to many life-saving cures and treatments. For example, the California Biomedical Research Association states that nearly every medical breakthrough in the last 100 years has resulted directly from research using animals. Animal research has contributed to major advances in understanding and treating conditions such as breast cancer, brain injury, malaria, tuberculosis, and many, many others. They also like to claim that there is no adequate alternative to testing on a living, whole-body system. Living systems like human beings and animals are extremely complex. Studying cell cultures in a petri dish, while sometimes useful, does not provide the opportunity to study interrelated processes occurring in the central nervous system, endocrine system, and the immune system. And conditions such as blindness and high blood pressure cannot be studied in tissue cultures. Also, computer models can only be reliable if accurate information gleaned from animal research is used to build the models in the first place. Even the most powerful supercomputers are unable to accurately simulate the workings of complex organs such as the brain. And finally, they claim that animals must be used in cases when ethical considerations prevent the use of human subjects. When testing medicines for potential toxicity, for example, the lives of human volunteers should not be put in danger unnecessarily. But what about the case where the scientific knowledge to be gained is minimal, while the cause of animal pain and suffering is great? Is scientific research on animals always justified by the virtue of the fact that it is scientific research? These are the questions that arise by those that are against live animal research, and these are very valid concerns. One argument is that there have been many scientific studies in the past that resulted in no new cures or treatments for humans, and yet many animals were used or even killed as part of the research. In fact, most research done doesn't directly result in a cure. There are many flops before a treatment is found or something useful comes from all that work, and possibly pain and suffering in the case of the animal. Which leads to another argument, that animal testing has the potential to cause pain and infringes on the animal's interests. Whether the pain and infringements on the animal's interest is getting poked with a needle every day, being injected with a vaccine, having implants put into their bodies, being fed food that may have adverse effects, or just being in confinement, unable to express normal behavior and enjoy life. It is not hard to see how animal testing infringes on an animal's interests. Another position of anti-live animal research is that there are now alternative ways to do the research. Alternative test methods are methods that replace, reduce, or refine animal use in the research and testing, a concept first described by William Russell and Rex Birch in their 1959 book, The Principles of Humane Experimental Technique. Their concept states that we must A. Replace animals by substituting non-animal systems, such as computer models or biochemical or cell-based systems, for traditional methods, or replace one animal species with a less highly developed one. For example, replacing a mouse with a worm. B. Reduce animal use by decreasing the number of animals required for testing while still achieving scientifically valid testing objectives. And C. Refine animal use by decreasing or eliminating pain or distress in animals or enhancing animal well-being. Alternatives can consist of cell and tissue cultures or computer models, which are cheaper, quicker, and sometimes more effective. And lastly, while many may make the argument that animals are protected by the Animal Welfare Act, the fact is that most livestock species used for production and many, if not most animals used for research do not fall under the parameters of the Animal Welfare Act, and therefore are unprotected from being taken advantage of.
So, in conclusion, what should be done to address the ethical problems? Well, for starters, we have some alternative methods that may replace the need for animals in the future, such as computer methods, cell and tissue cultures, or the use of lower vertebrates. Alternative methods do provide cost and time efficiency, and they require less manpower. As far as computer methods go, we would use computer-generated simulations to predict effects of chemical or potential drug candidates without the need for animal dissection. CADD is a computer software that's used to tailor-make new drugs for specific binding sites, and then in the final stages, animal testing is done to obtain the confirmatory results that we need. So hence the total number of experimental animals is lowered. Then we have cell and tissue cultures. These involve the growth of cells outside the body in laboratory environments and can be an important alternative for animal experiments. The cells and tissues from the liver, kidney, brain, etc. can be removed from an animal and can be stored, kept outside the body in suitable growth medium for a few days to several months or even a few years. Then we have the use of lower vertebrates. This is an attractive option because of the high genetic relatedness to other mammals. This means that there are less ethical problems involved in the experimentation because of the use of lower vertebrates. Zebrafish, for example, are an attractive option because of their transparency during early development. This allows for easy visual access to internal anatomy. They can be used for the detection of various toxicological studies of chemicals and pharmaceuticals. So which method is the most ethically acceptable? While the alternative tests that exist today can greatly decrease the number of animals needed for efficient test results, but it's hard to eliminate them 100% without jeopardizing all of our medical advances. Because there will always be a demand for these medical advances, it's very important that we choose a method that produces the best results medicinally, but also limits the amount of animals being tested on at the same time. And the most important thing
is that the animals are free from pain and stress as much as possible.